Our next thematic block is From Seeds to Supper. We're going to dive into the role of the seeds banks in transforming, of course, our agri-food systems. So how can seed banks best support and underpin the efforts to transform agri-food systems? And we're going to start again with a little video. The president of the Alliance for a Green Revolution in Africa, Ms. Agnes Kalibata, has sent us her words.
Excellencies, ladies and gentlemen, thank you for having me today. I'd like to commend the Global Crop Diversity Trust, or Crop Trust, under the leadership of the Executive Director, Stephen Smith, for putting this conference together. As AGRA, we recognize the work that Crop Trust does around the world. I'm especially proud of the Crop Trust's work with national and international Genebanks, and support to the Salzburg Global Seed Vault, which I visited in 2019 in the Arctic Circle. I also say this because seed is the foundation of all food systems and has been at the center of AGRA's work and strategies since its foundation. Indeed, all our 3 strategy periods since 2015 have had seed and seed systems at the center of what we do. To us, seed systems and crop diversity are closely interconnected and play a vital role in food security, agricultural systems, and biodiversity conservation. The maintenance and enhancement of crop diversity is essential for long-term food security and resilience, especially now in the face of climate change, environmental challenges, pests and diseases, and all the challenges we see today. But it's also essential to strike a balance between modernizing seed systems on one hand to improve crop performance and conserving traditional crop varieties to protect genetic resources and traditional knowledge. It is now clear that in the interest of food security, farmers in dry areas are encouraged to diversify their options through the cultivation of crops that can survive adverse weather, such as sorghum, millet, cassava, and fonio. Legumes like cowpeas, green grams, and chicken peas are also great options for protein. Seed banks allow us to maintain genetic sources and resources around these crops. Yet it's not just the dry lands that we are worried about, all that are under pressure. The 2021 Food Systems Summit that I stewarded on behalf of the Secretary General recognized the fragility and dysfunctionality of our food systems and spotlighted crop diversity as among the areas that require urgent transformation in agri-food systems to curb the loss that we see. We recognize that a lot more work is required in this area through investments in research to develop the right crop varieties for the ever-changing ecological environment. Hence, it's really critical to have good partnerships with the CGIAR. We also need investments in the maintenance and conservation of crop species through, for example, functional gene banks at national and regional level. This will reduce risks of gene erosion and thus the need to highlight the critical role that the crop trust plays in crop diversity. Finally, we must all recognize the centrality of governments in all transformation and work with them to strengthen their national seed systems, establish frameworks for promotion of climate-resilient varieties, and support their engagement with farmers through extension. All this will help us to quickly reach our global crop diversity goals. I have a lot more to share with you and to say, and I want to thank you for putting this meeting together. But let me conclude on this one important note. Africa as a continent has survived because we have significant diversity on this continent. We want to maintain our crop diversity, and we believe the crop trust has a huge role to play. Therefore, I want to encourage and call upon all of us to seize this moment and promote the role of crop diversity in advancing inclusive, resilient, and sustainable food systems. This is the opportunity in our midst. Thank you.
Thank you, Agnes, Kalibata, for sending us the video and, of course, for sharing your insights with us. Now, diving into our next panel debate, I warmly welcome our next panel moderator. She is the Global Genebank Partnership Coordinator at the Crop Trust, as we know, an organization dedicated to conserving and making available plant diversity. So I'm happy to introduce you and welcome you now on stage. With us is Janny van Beem. Janny, the stage is yours. Thank you.
Greetings, everyone. Thank you for coming back from lunch. That is already a good crowd. We are in session 3, the role that seed banks need to play if we are to meet the demands that are put upon us for agri-food systems in the future. So we have a brilliant panel. As you can see, the stage has changed. And I'd like to take a couple of minutes to let you know what the flow will be of our presentation, because it's slightly different from what you have seen in the past. So in the first part, we will learn about the complexities of a gene bank business. So everybody thinks, how hard can it be? You put the seeds in a freezer and then what? Distribute. It's not that at all. We will see what it takes, the hard work that it takes to take these seeds and create a form of art, if you wish, in the distribution, in contacting breeders, in making it available, in characterizing it. In the second part, we will be talking about the future. And I think it's why we're all here. We have a brilliant speaker that will set it off. That will be our third presentation. And we will have interventions by several people that will ultimately give us what we need to do in the next 10 years to be positioned as seed banks to transform agri-food systems. What is it? What is our wish list? And what do we need to put in effect? So in effect, it will be a strategy, what you bring forth today. So with that, I'd like to welcome our first speaker, Dr. Marco Wopereis from World Veg. Marco? Yes? So Marco is the Director General of World Veg Center. He's an agronomist with a doctorate in tropical agronomy from Wageningen University with global expertise in agricultural science and management. The floor is yours, Marco.
Thank you very much, Janny. Good afternoon, everyone. Very nice to be here. A big thank you to the organizers for having me. It's a big honor to kick off this particular panel. And keen to talk about sowing seeds, meeting needs. Sowing seeds of a wide diversity of food crops, obviously to meet the needs of current and future generations. And central in my talk are seed banks. We talk a lot about it already this morning, but what are seed banks? Really, these are places where we store seed for a long time under low temperature conditions. If we do that well, these seeds can be viable for decades, even 50 to 100 years. That's very important because biodiversity is declining, as we heard this morning. So it's a safe place for these seeds. And if we do that well, if we manage these seeds well, we can also send them out and test these seeds in different environments. And we may be surprised to find seeds that do extremely well under high temperature conditions, do well in terms of pest and disease resistance, etc. So it's like a treasure box. And sometimes I feel we've only scratched the surface of this treasure box. And you see here the glimpse of the seed bank in World Veg in Taiwan, and also a small collection of cucurbit family, just a snapshot of the beautiful diversity we have in that seed bank. Just also to underline the importance of seed banks, we are fortunate to have had a very visionary gene bank manager in the 1990s. Her name was Dr. Engler from the Philippines. And she collected seeds in 10 Southeast Asian countries. 12,000 samples she collected in about 1 to 2 years. And we are now in the process of bringing these seeds back home, because these seeds have often not been stored very well in these different countries. They're no longer viable, and we're bringing them home. And actually, if you would now go out in the field in these countries, like Philippines, Thailand, Malaysia, perhaps won't even find them. Right? So yeah, vegetables. It's a wonderful job I have. It's colorful. It's complex, right? So we work on global favorites like tomato, pepper, onion, but we're also very keen to work on other vegetables that people sometimes call neglected vegetables, traditional vegetables. I've learned a new term last week when Professor Lindui Magella Sibanda was at our center in Taiwan. She calls them opportunity crops. Okay, I think that's much better because these opportunity crops have so much potential in terms of nutrition, but also in terms of sturdiness, climate resilience. So we really have to also focus on the lesser known vegetables that are out there. And in general, bringing vegetables in food systems will bring resilience because farmers will have just more options to have income and more regular income because vegetables can be harvested multiple times. So it's just giving them regular paychecks, if you like. So yeah, wonderful diversity. So our seed bank in Taiwan is seen here on the left. So it's the biggest collection in the world in terms of vegetables, and we store there 62,000 accessions, we call it, means samples, seed samples, 62,000, covering 330 species, coming from 155 countries. And that has been built up over time. So it's a fantastic resource. We are very careful with it. We are also backing up that collection because imagine there's a fire or an earthquake in that particular seed bank, we have a big problem. So we are actively backing up this collection to other sites outside Taiwan. One is to Korea, and the other one is, of course, in Svalbard, and you see the beautiful global Svalbard vault there to the right. We are currently at 51%. Back up in these two locations, and by 2025, Martin van Zonneveld, our head of genetic resources, sitting there, promised me that it will be 100%, and it's essential. Okay. Now, all this well and good, but we talked about this declining biodiversity, and in the UN Food Systems Summit in 2021, World Veg with partners, including people here, and of course Global Crop Diversity Trust, rang the alarm bell because of this declining biodiversity. You're losing options. We must do something. So this is a paper you can still download, and the bill is $250 million for 10 years, okay, to do something drastic about this. So it's difficult to say how fast biodiversity of fruit and vegetables is declining. I don't really know. But if I look at what's on this slide, I'm worried because it shows that there are large gaps in what we have in our seed banks, right? So a quarter of the about 1,100 recognized vegetables is not conserved at all in seed banks. And perhaps the rest, we have 1, 2, 3, 4, 5 samples somewhere. So there is a lot of work. Also very important is that what we call wild relatives of vegetables are also not conserved very well. And you think, wow, is that a problem? Well, if you think of tomato, a very important disease in tomato called yellow leaf curl virus is a big problem, but because of resistance found in a wild tomato in Chile, now we don't have a problem with that virus, okay? So producers don't have to deal with that problem anymore because of that resistance that was brought into cultivars. So don't underestimate the importance of wild relatives. So the first step in implementing the Global Action Plan is through our TAVI, Taiwan Africa Vegetable Initiative, where we're actively trying to rescue biodiversity in hotspots. So we identified 6 hotspots of vegetable biodiversity in Africa, and we are sampling in 4 of those, in West Africa, East Africa, South Africa, and Madagascar. And over the 2 years, we're working with national partners in these countries. We have now collected about 15,000 samples. This is tremendous, but it means also a lot of work because you can collect them, but then you have to really multiply them and understand whether there are any duplicates, and then you have to dry them and store them in a seed bank, and then you must use them as well, otherwise there's no benefit, right? A lot of work still to be done, even within this context, and then I'm not talking about the other hotspot, I'm not talking about Asia, I'm not talking about Latin America, Central America, where there's also a lot of work. So another very important paper written by Martin called Forgotten Food Crops in Sub-Saharan Africa for Healthy Diets in a Changing Climate. What people did here is to look at, I think, 150 or so forgotten food crops in Sub-Saharan Africa and look at what potential they have in a changing climate and in providing nutrition to consumers. And in the end, they came up with a short list of about 48 of these crops. And we looked at cereals, roots and tubers, fruits, vegetables, you name it, nuts, right? So in the end, about a short list of 48 of these are there. And vegetables and pulses are among them to create double wins for climate resilience and nutrition. Think of amaranth, African eggplant, jute mallow, mung bean, etc. So these are all opportunity crops that have great potential. So in terms of rescuing biodiversity, we should put emphasis on those crops because it's going to be difficult to do that for 1,100 different vegetable species. Okay, many people already this morning said seed banks are not a museum. That's clear. So we need to connect with users. So we do that in several ways. So we have an online system where you can, as an interested party, click and order seed. And so over the last, whatever years, 50 years, we have sent out 700,000 seeds around the world, as you can see on the map to the left, from our seed bank, which I told you has 62,000 samples covering 330 species from 155, I think, countries. Meanwhile, we're also investing in Africa, in a seed bank there, since 2013 in Tanzania, in Arusha. And this is currently being upgraded to also have -18 storage capacity. That will be a very good thing. This is rapidly growing. I told you we collected 15,000 seed samples. Eventually, they will get in here. Currently, we have 6,000 seeds, mainly from African traditional vegetables. The opportunity crops, if you like, covering 100 species and coming from 49 countries. Here also, over time, since 2013, we have sent out roughly 450,000 samples, mainly in seed kits, to interested parties, NGOs, farmer organizations, etc. Another way we connect biodiversity with users is through the private sector. So in late 2016, we established a consortium under the umbrella of the Asia-Pacific Seed Association. Currently work with 51 seed companies in the Asia-Pacific. And these companies have an advanced look at our breeding lines, which are based on the genetic diversity in our seed bank. So this way, we bring more genetic diversity through our breeding lines to these seed companies that operate. Important because in the Asia-Pacific region, 1 out of 3 farmers will buy seed. 25 to 30% of farmers buy their seed from seed companies. So the only way to get our work in the field, you have to work with the seed sector, and you see the priority crops on the screen: tomato, pepper, bitter gourd, pumpkin. Bitter gourd is my favorite. That's the one to the right below. It's wonderful if you eat it with salted egg in Taiwan. Okay, so we have a little sister in Africa that is growing also, was established later. We have currently 60 entities, I would say, not really companies, farmer organizations, more informal undertakings that have the ambition to eventually perhaps start breeding vegetables for different niches. So here there is a lot of emphasis on evaluation of what we have in hand. Whereas in the Asia-Pacific region, we work with very mature seed companies. They pick up our breeding lines and continue the breeding process, right? So here it's a lot about evaluation and selection. The list of priority crops is larger. Another example I wanted to give is on mung bean. Mung bean is probably the crop that we had most success with in terms of impact. And we work there with the public sector, national research institutes. So we have a wonderful collection of mung bean, 8,000 accessions. But if you want to evaluate 8,000 accessions in 14 countries, it's going to be a little bit difficult, right? So we constituted what we call mini core collection of about 300 accessions that cover the entire diversity of these 8,000, right? So that's manageable, 300. So you see a picture on the screen of that 300, that set of 300 being evaluated in India. And at that time, we had a problem with mung bean yellow mosaic virus. And you see that some of these accessions in the 298, to be exact, lines do very well. Others succumb to the disease. So by putting that mini core in different places around the world, we start to find wonderful characteristics that we can work with, either directly or in the breeding process. So another last example I want to give is on seed kit distribution, bringing diversity into farmers' field of different varieties through seed kits. Sometimes these seed kits are made by World Veg, sometimes they're actually made by a seed company that we work with. And through these seed kits, we work with NGOs, governments to bring seeds into a home garden context, for example, or into smallholder farming. And some of the most popular traditional vegetables that we work with, you can see on the screen. Amaranth was mentioned already this morning several times, right? It's a C4 crop, very nutritious, very interesting. Jute mallow, spider plant, African nightshade. Okra is also very interesting. On the picture, you see a champion farmer from Madagascar that has worked with these seed kits, and she is showing off here her Ethiopian mustard crop and her amaranth crop. Okay, few words about complexity, okay? Yeah, I told you we have a seed bank, we have actually two, and the seed bank has 330 species in it. And so the complexity is really quite mind-boggling because every one of these species have different growth characteristics that you have to understand. And the seeds may have to be treated differently. So we have 8 curators. A curator is somebody who takes care of vegetables, let's say, and 8 of them divide the pie of 330 species. So it's a very complex undertaking. And yeah, to send out seeds means like, okay, seeds arrive, you got to multiply them, you got to clean them, you got to dry them to low soil moisture, you put them in -18, we get a request, oh, then we have to go into the fridge, the freezer, and get it out, start again, open it, and check it if there is no disease problem with it, phytosanitary permit, etc. Hugely complex undertaking. Voila, I see my light is orange. So, last slide, seed banks. I just wanted to put 4 C's on the table. Colorful. Colorful, that's wonderful because color with vegetables means healthy. Right? The more colors on your plate, the healthier your plate will be. Complex, I think that's pretty clear. Critical to address the climate, nutrition, and biodiversity crisis. Costly. To run these 2 gene banks costs us $1.4 million a year. $1.4 million a year, and the funding is all time-bound. And we're so lucky to have 1 or 2 very big supporters. But all the funding we have is time-bound, and we are grateful to start getting into long-term funding from the Crop Diversity Trust to help us, in particular, in Tanzania. Thank you very much.
Thank you, Marco. That was great and impressive work. Not only maintaining the quality in your gene bank, but also the many different actors that come into making your gene bank useful. Equally impressive is the evolution that the CGIAR has gone through in their gene banks in the last 10 years. And I'd like to invite our second speaker today, Juan Restrepo. He is the Global Director of Partnership and Advocacy, CGIAR, and also the Director General of the Alliance of Biodiversity International and the International Center for Tropical Agriculture. Welcome, Juan Lucas.
Thanks a lot. Thank you so much. It's great to see you all. I'm going to stand from here so you don't have to watch my back, Marco.
Let's see it here. Yeah.
And I really enjoyed listening to World Edge as an international research organization similar to the partners constituting CGIAR, with that mandate and a very important public good that you're managing. This conversation is very close to me because I'm not only here as CGIAR, but I have a long-standing work from the public sector, from our Colombian National Agricultural Research Institute, connecting all these plant genetic resources for food and agriculture. So what's being discussed here is very dear to me and critically important. So I will basically a snapshot of what means these 11 gene banks in trust collections that we manage on behalf of a society. We're talking about 3,000 species of 30 staple crops across 5 continents. A very large, and we will see some numbers, mandate not only to conserve but also to distribute, and we are a very important actor in this multilateral system under the treaty. And it also makes us very proud to see that this effort is focused mainly in sharing and exchanging with the Global South and in partnership. So crop breeding networks, small enterprises, small national institutes, not only at the national but also at the sub-regional level, and also work directly with farming communities that are critical, not only in conservation, but in connecting conservation to use. These are some of our numbers from our last CGIAR report, and as Marco says, it's a very complex business. We need to keep innovating. We need to keep doing research in terms of new standard operating procedures, new ways of dealing with diagnostics of sanitary health of the sample. So there is a lot of continuous work and obsession as well to enhance our performance, and the performance not only to manage these collections well, but also, of course, to manage them at the lowest per unit cost possible, because these are very costly and important operations. Gene banks are at the core of a lot of CGIAR's work, and this is just a way to show the genetic initiative side of things in CGIAR, and how all of our efforts in CGIAR related to breeding, to market intelligence, etc., use gene banks at a base, and how this work is very well connected, organically connected to our 5 impact areas. So it's not only around food security, but it's covering gender equality, climate adaptation, biodiversity, etc. So some highlights and examples that showcase that this is not just conservation, but use and learning more and more how to valorize this work. So in terms of breeding, there is a very interesting and valuable work for farming in terms, for example, for mining, unlocking, discovering alleles in maize, rice, cowpea that are connected to climate change adaptation from gene banks, and drought and salt tolerance, etc. And this is work that produces a lot of information connected to those accessions that can then become a very important input to breeding efforts, or also identifying materials that can be readily be used by farmers. There is also work identifying traits associated to aflatoxin resistance, for example, in groundnut, pigeon pea, and other areas. And the latest effort that I think is very interesting and tells you the potential that gene banks have is dealing with climate mitigation. And you know, ruminants, forage eaters are important sources of methane. So we are working now in looking at the forage collections and identifying those molecules, those compounds that are associated with lower emissions from animals. And then as we identify that, bringing those materials into breeding, so new forages that emerge from these breeding efforts when fed to animals mean those animals will be well fed and also lower in terms of their emissions. So there is a lot of potential of what can be done and what can be unlocked from gene banks. But there is also an amazing experience and possibility through all of that diversity to just identify materials that are ready for use, where opportunities are readily at hand. And there is beautiful examples. One is a cooking banana that's vitamin A rich, and vitamin A deficiency, especially in small children, is critical for development, and how those vitamin A rich bananas from the banks are being brought back to farming communities and well uptaking and solving important nutrition deficiencies. Or how we have, for example, identified yam bean varieties through participatory selection efforts with farmers in a number of parts of Africa. There is also this, and you mentioned some of this, Marco, this repatriation of collections. We hold these collections in trust, and they are the backup in many cases for farming communities. So it's great, for example, to see this year SIP repatriating hundreds of land races of tubers to 20 farming communities in Peru, as an example. Or how, in a similar way, Africa Rice helped repatriate a number of land races of rice to Chad and to help kickstart its national improvement program. There is also, and we'll hear from the Vault, Svalbard, how, and this is an amazing success story, how ICARDA, after what happened in Syria a few years back, managed to restore its collection in Egypt and Morocco, drawing on Svalbard and coming back to having a gene bank that's again sharing and meeting its mandate. And also to finalize this part, signaling that this is all about partners, that this is not a CG by itself, but this is understanding what capacities, especially in the national systems, are there, and also focusing our efforts and engaging directly, for example, with some disadvantaged NARs, so we can support their data management standards, etc., to help them raise some of their performance. Or how, again, our new learning on cryopreservation that's so promising for some of these species that we need to maintain are quite costly, is something that we also share in networks. So partners learn, or we learn together with partners. So there is a number of examples on that. And then to pin this down, I think CGIAR's footprint in the global system is quite important. And in terms of the treaty, 90%, and this needs to change, hopefully, but so far 90% of the exchanges, of the transfers of materials under the SMTA of the treaty is CGIAR material. And this is something that's great. We are very proud of that. But it's something that means that there are elements in the global system that need to be addressed as we move forward. So in terms of lessons learned, collaboration is key. We cannot work in isolation, and working with national programs is very important. Also linking, it's not the national public sector, but going one step down and understanding what local communities and farmers need and also can do in terms of conservation and use of these materials. There was a discussion this morning on opportunity crops, neglected and underutilized crops, where we have a lot of learnings from staple crops that can be transferred into neglected and underutilized species. We cannot start from scratch. That would not make a lot of sense. And also understanding that these crops need to be more and more part of the overall solution to meet SDGs and societal goals, and the need, therefore, to work on this area where also we have some experiences that we can share. Then the connection of ex-situ and ex-situ is very important. Gene banks are very critical, but we need to make sure evolution is there, that things evolve, that diversity is tested and withstands biotic and abiotic pressure over time. So we make sure we also provide for solutions in the longer term. Meaning we need to understand in terms not only of wild relatives, but also these farmers' materials, how we work with them and they are functional part of the system, and for this reason also how we functionally connect with community seed banks, or we, meaning national systems as an ecosystem, is going to be very important. Just to start closing on the way forward, there are some important challenges. We still lack a lot of information on what is there. We may be duplicating efforts. There is still a lot of lack of sharing of information that we need to work on together. The access and benefit sharing under the treaty is still not where it needs to be, and therefore, implies that things do not move as fast as society needs. So that's something where we need to work on. And given CG accounts for 90% of exchanges, so well, let's come with creative ideas and ways to see if that can be better connected to access and benefit sharing. And then there is, I feel, and this is not CGIAR, this is my personal opinion, there are gaps in terms of coordination and clear sharing of responsibilities in what I see is a plant genetic resources public system where you have FAO with the commission, with a specific mandate as an intergovernmental body, the trust, the treaty, CGIAR, and the NARES, and under NARES, understanding that there are some very large and important ones that need probably to have more functional and take on some of the responsibility, whether global or regional, and see how we optimize this. What do we, especially in a cost resource, resource scarce environment, what do we need to share, how we support each other so we work as an institutional public ecosystem and become more effective at meeting our aggregated mandates. And then just to finalize, basically there is also things we need to do quickly to make things work better. Digital sequence information is something we do not talk enough about, but it's limiting how countries really are ready and willing to share more and to work more in this area. So that's something we need to tackle, I would think, urgently. And of course, ensuring that we get increased understanding and investments, not only in agriculture, not only in research and development in agriculture, but in the gene bank business that's so important. And then, of course, trying to leverage and optimize. We have done a lot, we are doing a lot, but I think as we accommodate ourselves better in this ecosystem, how our infrastructure, technical expertise can be leveraged even further to strengthen capacities at the global level and of this system to meet societal needs. So this is future seeds that the newest gene bank of the system in Colombia that hopefully you can visit as Stefan did when he was present in the inauguration. Thank you so much.
Thank you very much, Juan Lucas. We appreciate the thought that you have given to not only showing about gene banks, but taking it a step forward, biodiversity into partnerships and also giving us lessons learned and solutions. That's excellent, and we will take those in mind. I'd like to invite two speakers now for a little round table that we will have. The first speaker is from Bhutan, Dr. Asta Tamang. She is the principal officer working at the National Biodiversity Center under the Ministry of Agriculture and Livestock of the Royal Government of Bhutan. Welcome, Asta, and please take a seat here. And our second guest is Dr. Daniel Kotey. He is acting director of the Plant Genetic Research Institute in Ghana.
Hello.
Daniel has successfully coordinated reviews of the international and technical capacity and leads the team that implements a quality management system within Ghana Gene Bank. Please come forward. So we have about 10 minutes to discuss. This section was about the complexity of gene banking and the achievements that we should learn from so that we continue to implement them in gene banks. So I'd first like to hear from Asta about what she considers are her basic achievements, her most successful achievements in the Bhutan Gene Bank, and some of the lessons that she has learned along the way. Yes, please use the microphone.
Long arm. I got a long arm.
Yes.
Thank you, Janny, and distinguished guests and participants. Very good afternoon. As many of you might be kindly aware, Bhutan is a very tiny country located in the eastern Himalayas. And since the operationalization of the Bhutan National Gene Bank in 2005, we have made concerted efforts and secured a germplasm of rich crop diversity, hidden in the deep pockets, deep valleys of the Himalayan kingdom of Bhutan. While the gene bank in Bhutan is relatively young compared to many gene banks around the world, there are about 3,500 accessions of germplasm from about 46 crop species. That are of importance to national and global agri-food systems. Now, if I may touch upon the utilization of the germplasm that are being conserved in the gene bank, we really look into promoting the utilization of the germplasm. There are different users of germplasm, the research and development centers, the universities, the farmers cooperatives, etc. If I may cite an example of utilization of germplasm from the gene bank, well, by the way, Bhutan is a carbon neutral country. In fact, it is a carbon negative country, and Bhutan is serving as a carbon sink for the global community, in fact. And despite Bhutan having very robust policies in terms of environment preservation and biodiversity conservation, Bhutan is not spared by the impact of climate change. And farmers are confronted with numerous impacts of adverse effect of climate change. And to name one is like drying and shrinking of water sources, drought, dry spells, etc. So the gene bank provided germplasm to the universities and to the research centers, rice germplasm, in fact, and they do the evaluation and select the climate-resilient varieties or the drought-tolerant varieties of rice, contributing to seed security and agri-food system. So Then, if I may, let me tell you about so-called buckwheat in Bhutan, a place called Bhutan in Bhutan. Bhutan stands at an elevation of about 2,800 meters above mean sea level. And the buckwheat used to be a staple crop of Bhutan until 1960s and 70s. But over the years, the cultivation declined. So at one point in time, some 12 years ago, when I went to collect germplasm for the Genebank, it was difficult to get seeds for the Genebank. And at one point in time, that was a staple crop. So we created awareness amongst the farmers, the importance of the crop diversity. And we formed a farmers group, the interested farmers, and provided them with the support, like in terms of developing different type of buckwheat products and marketing it. So that, and also we provided like support, a market outlet or the buckwheat outlet. Then over the years, we could see the impact that, multi-dimensional impact, after certain years. That was not seen, that was not there before. Like from buckwheat, it became a source of income generation. It provided employment opportunity to the youth. The success of this group actually inspired farmers of other communities, again, to form groups and cultivate buckwheat, which they used to cultivate before, but they had stopped. So they started cultivating, and it enhanced the production of buckwheat. And it created market demand at local level, at the national level, and international level for buckwheat. So contributed in the better agri-food system at all levels. Not only that, it sort of helped in reviving 3 C's, like the crop, the culture, and the cuisine. It also had other, impact in other dimensions, like it provided ecosystem services, because we see a lot of flowers in buckwheat, so a lot of pollinators, a lot of honeybees, and also there are indications that it sort of increased the honey production and all that. So a lot of impact, and it enhanced the farming system resilience, and well, if you still give me some more time.
Maybe 30 seconds, and then we give the floor to Daniel.
So let me tell you about the formulation of mutually supportive ABS mechanism in Bhutan. Based on our national need, and also recognizing the importance of the objective of the Nagoya Protocol under CBD, and also honoring the noble objective of the multilateral system of ITPGRFA, we formulated ABS policy, Access and Benefit Sharing Policy, and also Biodiversity Act of Bhutan, which do have a component of ABS policy, thereby harmonizing the Nagoya Protocol and also multilateral system of the CBD and the ITPGRFA. Yeah. So thank you very much.
Very good. Thank you so much, Asta. Thank you so much. Two very good success stories, and I think that's something to be learned about that. So next we will have Daniel from a completely different region, and he will talk about accomplishment in his gene bank.
Thank you very much, Janny, and thank you very much to my co-panelists. As has been said before, gene banks are not museums, and they need to connect with users to make sure that users have value in terms of access to genetic resources that they can use for their various needs. And as gene banks, we can only get to farmers or connect with users if the material that we conserve are alive and they have characteristics that the farmers can pick or select from. And so for us, I would say that our key success has to do with the implementation of gene bank quality management systems that ensures that our internal processes are maintained at a higher level such that we are able to deliver value to our germplasm users nationally and internationally. And nationally, principally, smallholder farmers that are subject to the vagaries of the weather and are at the forefront of climate change mitigation and adaptation. Then how we were able to achieve the success. We were able to achieve the success because there was local buy-in and local ownership. The Ghana Gene Bank was established in 1964, and for most of its history, we're focused on collecting and securing the germplasm ex situ. But we didn't focus much on our internal processes. So whilst we were conserving, we realized that the value of the germplasm that we had to farmers was not as high as it should have been because we're not focusing on quality processes. And so when the Crop Trust came to do an evaluation of the Gene Bank, they came up with recommendations. And based on these recommendations, staff of the Gene Bank took the pains to prioritize areas of focus. And once we prioritized the areas of focus, we now had a participatory process with experts appointed by the Crop Trust to develop quality management systems that we implemented. And because there was participation by our staff, there was ownership, and they knew exactly the value of each of the procedures. And so it wasn't like an imposition. And that has led us to now operate at a very high level to secure the genetic resources that we conserve for now and the future. And one of the significant things to note is that the Ghana Gene Bank conserves a multitude of crop species, right from tree crops, medicinal plants, spices, to roots and tubers, and other cereals and legumes. So our collection is very diverse, and it has a lot of value for climate change adaptation and for maintaining food and agricultural system diversity. And so for the lessons that it has for other gene banks that are intended, or that intend to also operate at such high level, is that first of all, there has to be context-specific development of procedures, and staff must understand clearly what each procedure is supposed to achieve. Once that is done, it's like they themselves would adopt these procedures and implement them to the benefit of all the users that access material from the germplasm. And by so doing, some of our unique crop collections that are conserved by national gene banks can be made available for farmers for sustaining diversified farming and food systems that also preserve our food culture. So thank you very much.
Thank you very much, Daniel. That was excellent. Definitely some lessons to be learned as well. And I liked the way that you said quality inside the house. If you fix your house, it's like a rolling ball. It does roll back, the quality does roll back to users, to breeders, to farmers, to researchers, and everybody else. So that is something that we need to remember. We're on to the second part of our session, and that is the part where we look to the future, and we want a to-do list. Things we should be doing, and as somebody said yesterday, not even today, but yesterday, we should be doing these things to position gene banks to be able to meet the challenges of the future. So I'd like to welcome Dr. Susan McCouch to the stage. She will be giving our talk. First of all, start with the futuristic, and then we can invite the rest of the panels. Susan is the Barbara McClintock Professor of Plant Breeding and Genetics in the School of Integrative Plant Science of Cornell University. She is known for developing the first molecular genetic map of rice and for her key and sustained role in turning rice into a model for genetics and breeding research. Welcome, Susan.
Thank you.
Being in her lab.
Well, I want to say thank you for inviting me and for this invitation to join this esteemed panel and to meet many of the gene bank managers at this meeting that I had not met before. They're an inspiring group of people, and my comments here are a little bit from the sidelines because I'm not a member of either the CG or the Gene Bank Consortium here, but I have had a long association, and I care deeply about this topic. So I'm going to talk a little bit about how we can empower our seed banks, be they national, international, or local community seed banks. Combat is not maybe my favorite word today, but we are dealing with some very serious threats. And I'm just going to try to focus on these questions that Janny asked, which is, what can we be doing so we're prepared? So do I have a... Yes, it's
here.
Yeah. Okay. Yes. So I would say, in the spirit of that lovely speaker who came before me who spoke about the culinary delights, I would almost ask you to close your eyes and imagine the diversified agri-food system that would be in your dreams of what you might think that we as a planet might be aspiring to. I've given you two of my favorite concepts here, but as you can see, I come from a very green place where there's plenty of water. And some of you may dream of diversified systems that don't look at all like these. But the point I'm trying to make here is that the diversified agri-food system, as we envision it, as we aspire to, it provides resilience against climate change, nutritional deficiencies, and the challenge of livelihood. And I think we've just heard some indications from others about how this might work. These are systems where we may be talking about seeds and crop varieties as inputs, but these inputs are much more than biological reagents. These, in fact, seeds and these varieties contribute cultural, social, and political meaning in the context of a traditional agricultural system. We have, at the moment, an international system that emphasizes these two different kind of concepts of seed conservation. And you'll see that along the left and bottom is a blue rectangle. Everything's quite linear. In the middle is a sort of yellowish, more circular diagram representing the formal seed system and the informal seed system. And the point I'd like to make here, if I... Oh, yes, very nice. Does this show on? Oh, cool. Okay. The international seed systems, the international gene banks sit here, completely disconnected in this diagram, at least, with the farmer-centric seed systems that are here. They, however, are connected in very important, both formal and informal ways, with national seed systems which have a more direct relationship with the community seed systems. Now, as I understand it, in the 1960s when the CG was formed, and I think this is important to understand historically, there was an agreement with the governments of the countries whose germplasm was going to be stored in the international gene banks, that the international gene banks would not, in fact, be involved directly with the farmers in those countries, because it is a very culturally, socially, and politically sensitive domain. And so I think this is why this is envisioned this way. We may want to think about how connection and connectivity might be improved, but you see that this system exists sort of outside, and here we have the community seed systems that are much more directly connected, and they're very farmer-centric. They have household use, community seed exchange use, and then local market, and of course, commercial use. So one of the issues that I'm going to be pointing out today is that I think we need to strengthen the relationships that exist between these formal and informal seed sectors. And they're both strong, and in some countries and for some crops, one is stronger than the other. But where they're both strong, leveraging that strength is going to help us deliver a larger array of diverse opportunities to the farmer or informal seed systems. It's also going to help, I think, the farmers, so the farmers would access greater diversity, but also the gene banks would be better able to reach the farmer and to find out what in their collections, is there water? Can you just pour me some water? What in their collections might be most useful? So I just want to remind everyone that when we ask where crop diversity comes from, we're not always talking about crop diversity that's very important, may even be the staple food within a certain country or region. Thank you. The origin of that diversity may come from far away. I'm sure many of you are familiar with this wonderful paper by Kauri et al. Shows many of the major crops of the world, where they originate, but where they are found today is shown more like in this diagram, where the major regions of the world are completely interconnected. So today, our crops, the crops we eat, most of the dietary opportunities that we have are not derived from crops that are local or indigenous to our own regions of the world. When crops come from another place, they come because of human migration, they come because of collecting trips or plant exploration missions, and they also come sometimes by mistake. They tag along with a shipment that may be shipping wood, furniture or something. But the point being that when they arrive in a new location, there's always a process of adaptation. Most crops or most crop varieties don't come to a new place with a complete adaptation. They go through a period of adaptation by either, depending on the time frame, traditional farmers, back when many of the crops from the Americas came to Africa, for instance. In this case, I'm showing the case of maize. It arrived in Africa after the discovery of the Americas by the Europeans. It came to Africa, and it is now the major staple in many parts of Africa. They have their own land races. There was a lot of selection. It occurred very early. And so in some ways, historically, maize is an African crop. It didn't originate there, but it has been there long enough that they have land races that are recognizable as African land races of maize. We have similar examples for almost all the major crops of the world, which is what this diagram is. My point is that the community seed systems connect farmers not only with crops that are indigenous or native to their areas, but with crops that came from elsewhere and have become important. And we have to make sure we keep this in mind. So I'm going to talk a little bit about the future now when I speak about the possibility of, again, redistributing germplasm across the world, regionally or interregionally. And the gene banks are in a good position to think about how this might be done. They can use all kinds of interesting machine learning technologies to predict weather and climate patterns, to see what grows well in one part of the world that might grow well in another, in a climate that is still a predicted climate where we don't yet know. But if the farmers were able to get some germplasm that's predicted to do well in their new environments or their evolving environments, they might be ready. They might backcross it into material that's already adapted, but that would help them, I think, prepare for a more diverse genetic base as they go forward. So the question is, what variation will be needed in the future? And if we knew that, boy, we would be in a good position. But we don't. And our predictions are based on, well, honestly, all predictions are based on the past. That's how we learn. We have an accumulation of data that comes since the time we're born. We learn to expect one thing based on a trigger that happens to us, and we make predictions as human beings. We've learned to train machines to make predictions based on the data that they have. They will use that data to predict patterns about what's likely to happen in the future, make predictions, and those machines may make predictions about, for instance, climate variables that the human mind is incapable of really managing, but they're still predictions, and they're still based on data that we have today. There's nothing magic coming down to tell us what's going to happen. But I will just use this slide to say I think we've got to think very carefully about all the many ways there are of learning. And we don't learn only in one domain, siloed in our own domain. I think the opportunity we have going forward is to try to mix and match across domains as much as is reasonable, as much as curiosity drives us, and as much as the infrastructure and educational systems allow. So we have to build, I think, from experience and traditional knowledge. The farmer in the field is going to come at it with a deep and profound understanding of how crops behave in the environments in which they're familiar. We can use science, and you'll see here's the young me developing the first genetic map of rice, and boy, I thought I had that predictive tool in hand. I thought that was really going to help, and I still think genomics is foundational for making predictions. And then we have what we call computational prediction, and we are all interacting with that all the time, whether we know it or not. I would say that what we want to do is to integrate across these different ways of knowing and predicting, and then testing the predictions in the ways that seem most appropriate for the beneficiaries or the users that we're trying to serve. So I will just start. You have heard from all of the people up here about their gene banks and the complexity of managing a gene bank, especially if you have 100,000 accessions or 62,000 accessions or whatever it was. So the question comes up again and again, which accessions should we be thinking about deeply and evaluating and optimizing and sending around? And I have 3 suggestions here that I think are reasonable for gene banks, and in fact they've been mentioned by every one of the people up here previously, but in slightly different contexts. We start with a simple menu and we build out from what we learn, because we're going to make predictions based on what we learn from this simpler menu. We develop core collections. That's a plural because you might have a wild species collection, core collection that's different than a landrace collection. You might have a collection of elite varieties that are adapted to certain regions. You want to use the fig selection method, where you're using the intersection between data that you happen to have on those varieties in your gene bank or traits, and with the geographic and agroclimatic information that you have from your passport data. You also might want to make trait-based subsets if you're a gene bank manager, because you know that there's a group of farmers that really needs drought-tolerant maize, or that really needs submergence-tolerant rice. And these things can be accomplished with a mission if there's a focus. And so creating trait-based subsets a priori can, based on a trait prioritization, and I have here prioritized traits based on farmers' needs, based on who it is you're trying to serve, and have them ready to go. Now, what would be involved? Well, you'll want, essentially, if you create these collections, probably with the help of some genomic data, as well as the passport data, as well as the trait data. Again, we integrate across ways of knowing. Work with community seed banks to amplify the seed stocks and identify farmer-preferred varieties for immediate use. There are a lot of ways to do this. There are participatory methods that are going to be essential to figuring out which of the varieties in your, let's say, in your core collection or in your designated trait subsets are most appropriate for use. Develop partnerships and collaborations. We've heard it. Everybody is emphasizing the same thing. I would say to rigorously characterize those collections, those simple collections to start, and to augment interest in that material on the part of many of the potential participants or beneficiaries, as well as to provide an essential experiential learning opportunity. Because while you grow out and characterize your material, if you do this in groups, you will learn what's important to somebody else, what the eye can see when it's a trained eye of a farmer or a breeder, how to use maybe a cell phone to take images and use the images to then put that in your gene bank, and how you can then do image analysis and you can move through a field much more quickly. Or how to use drones, if you have them, and do longitudinal growth and see how actual plant varieties increase in vigor and grow to cover the ground over time. You can do many different types of things, that opportunities for learning. So you establish some kind of inclusive community, however you need to define it, and it differs for every one of the crop communities. But I think it could optimally include farmers, breeders, researchers, and fellow or sister seed bankers, okay? So seed banks from other systems, national, international community. You leverage opportunities during the time that you're characterizing through this active experiential learning. You leverage opportunities to train a young generation of students or young professionals. Engage with universities and places that are training people, and be part of that training process, and they will be yours forever. This is a very important point. Get a young professional involved early in a meaningful pursuit, in a team where their contribution is valued, but they're not expected to be an expert in everything. And they will understand a way of working, and they will understand the mission. I think it's a way of recruiting the next generation of leaders, and we need them. Bring people together through these projects, but also meetings, workshops, congresses, summits, and field visits. Essential is the field visit. And I would say build cohorts of young, open-minded people, because those are the experiences that I think have shaped most of us here on this stage and many of you. I don't think you joined the crop germplasm conservation and utilization family if you aren't infected young. So I think most of us probably got that virus early. And then there's a whole dimension of working with breeders to develop populations for further genetic improvement, use of participatory approaches, and establishing very well-structured participatory breeding initiatives that are expected to give rise to varieties that are going to be useful in the immediate sense, as well as to create a cauldron of opportunity for continuous selection going forward. And I don't have time to talk about my favorite subject, which is breeding. So I eliminated all the slides on breeding. I think the idea of establishing a pipeline for innovation, if you're a gene bank manager, you should have a pipeline that claims innovation. And I don't know how anyone in this audience really thinks about innovation, but I've put a few of my thoughts down here. If you empower innovation in and for rural communities, you'd be rediscovering traditional crop varieties that are suitable for cultivation now. And as I was talking to some of you over the last day or two, I learned that in fact, some farmers don't know that the gene bank has the variety that their grandfather used to plant and they haven't seen since they were a little boy or girl, and it comes back from the gene bank and they recognize it. And probably in the home, the culinary experience is similar. So I think that sometimes we forget that rediscovery of something that was traditionally ours is in fact an innovation for the new generation who didn't know it existed. I think that's an innovation. And if we collectively can empower that pipeline, that will be an innovation pipeline. I think participatory strategies for both evaluating germplasm when it's still in the breeding stage and varietal testing toward the end to find out what really works for different communities. Once you become a participant in a project like that, again, it's transformative. So I think it's an innovation at the community level. I think adoption of new crop varieties is fairly self-evident. Crop varieties are innovations, they always have been, and new management strategies that sometimes have to go along with new varieties. And then my last point here is the discovery that gene banks not only collect and conserve, but also give back is one of the transformations that we have to work toward. And I think it truly would be an innovation if people understood the gene bank is here to give back, but it has to be able to give back the appropriate material. It's not a thing of just showering people with random diversity. Now, along with that, I think we have to examine the policy framework that affects these innovations, because the policy framework has been confusing and will continue to be so. Somebody mentioned, I think, that DSI is on the table. I love genomic data. I think it's the most powerful thing that a gene bank manager could have at their disposal in thinking about how to giving a framework for organizing their material. But it's a contentious issue in the international community. It needs to be addressed head on, and I know that we're all working on it. I just put that out. Do farmers have the right to register and sell seeds of traditional crop varieties that have been purified from gene bank accessions? I can read and read and read. I can't understand honestly exactly how to answer that question. I think that's going to be important to clarify, because farmers who receive traditional varieties from gene banks select on the heterogeneous populations they get from the gene bank and then call it a new variety. I don't know in different countries if they can register that variety and sell seed when it's actually directly out of a gene bank. But if we're advocating that they use this material, they need to be able to make a living on using it and amplifying it and the work that goes into it. Who is, in fact, the inventor of varieties that emerge from successful participatory breeding projects when there are multiple partners? Problems that are not, you don't want to wait till you have a new variety to address that question. And if participatory plant breeding varieties derived from gene bank parents become the foundation for a successful regional business, I could imagine that a community seed system and a group of of very interested farmers could be proactive in this. They could engage in a pipeline of innovation to generate new materials for their own communities. Would they be required to contribute to the benefit sharing fund? Because now they're a business. Interesting question. Okay, I don't know how to answer it, but maybe others do.
We have 2 minutes.
Oh, okay. So this point has been made and maybe should be made again, is that gene banks are in fact both resource and information centers. The information about the plant genetic resources enhances the value of their holdings, and it's the only way that we can actually, as users, find the diversity that we seek. But it's also important to realize that that information component, it may be, again, at various levels, uncovers hidden variation that a user might not even think to look for. And so information is a discovery as well for the user who comes in and interacts with an information resource. Information about plant genetic resources is often dispersed in public databases. This goes back a little bit to the DSI issue, but the fact is that genotype, phenotype, and ecogeographical adaptation, as well as nutritional value, generated largely now through high-throughput metabolomics and other kinds of data generation, is very powerful, even though it's not attached to a particular seed stock, because you can use, again, your predictive abilities to find relationships among data that can be leveraged for insight and inference, even if you don't have access to the genetic resources from which that information was derived. Many examples of that. And gene banks have a responsibility, I believe, to invest in data and information management systems. That's what they're there for, and that's the only way that they're really going to emphasize use. So that will enable more efficient and cost-effective management of their own operations. It will facilitate sharing of information and plant genetic resources with the diverse user communities we've discussed, and it will support informed decisions about future deployment and use. And I'm going to stop because I think I have run out of time.
Okay.
We certainly need to safeguard these collections. The point's been made, and it involves a lot of very, I think, mundane and traditional investments. I will just say that we also need to think about developing emergency seed amplification and distribution networks, because when disaster strikes, we want gene banks to be positioned to respond. And the only way they'll be positioned to respond if they start interacting a bit more among themselves and making the information about their collections more visible. So I do think that, I put it in the middle here, when searching for a novel PGR, plant genetic resource, people turn to gene banks, and they like to look through. But when an emergency strikes, what is familiar and at hand matters most. So if we've already got new sources of diversity in the hands of farmers in areas that might be stricken, those farmers are going to be in a much better position to help respond. I will say the BOLD project is making very significant contributions to strengthen and upgrade gene banks globally, but a lot more funding is needed to keep them not only intact and operational, but able to do what we hope they'll be able to do going forward. And my last slide is about communications and public awareness. Multimedia communication strategies are critical. Our job is not just to anticipate what we need to do as a community, but how the public sees us, how our user communities see us, and whether people trust us. So I think that these communications tell stories, they document impact, they raise public awareness about the importance of gene banks and crop diversity. They certainly help with the adoption of new approaches and technologies, be those technologies new crop varieties or anything else. But that requires trust and a shared framework of understanding. The storytelling helps to create a shared context for understanding. And I think the BOLD project is doing a marvelous job with communications, and many of you have probably been reached by what they're doing. So my general philosophy here is, these are my take-homes. Invest in excellence. We have some excellence right up here. I am so proud to be up here with this group of people. Reward success. Provide flexibility to encourage new kinds of partnerships, funding strategies, and project ideas. nucleate critical mass to stimulate ideas, attract funding, and maintain diversity of our teams as well as of our germplasm. Leverage existing strengths and build on them, connect existing gene banks, and finally coalesce around big problems that require novel solutions and engage with, I think, universities, that's my pitch, because we really, all of us together, need to provide opportunities that will attract young researchers to our cause. Thank you.
Thank you very much. Thank you very much, Susan. I'd like to invite 4 speakers to the stage. The first is Dr. Karen Mapusua. She is the Director of the Land Resource Division of the Pacific Community in Suva, Fiji. The first woman to hold this position, and she provides technical and scientific support to the Pacific countries and territories on all aspects of agriculture and forestry. Please sit. Our second guest is Dr. Lise Stephenson. She's the Executive Director of the Nordic Genetic Resource Center, where she leads the organization and heads Norgen, a joint gene bank and knowledge center for genetic resources in the Nordic countries. Our third guest is Dr. Anthony Okere. He is the Acting Director of the National Center for Genetic Resources and Biotechnology of NACRAB, an Acting Register of the National Variety Release Committee in Nigeria. Last but not least is Catherine Hazel Aguilar. She is the concept developer at the Leipzig Institute of Plant Genetic and Crop Plant Research. She works in the EU-funded PROGRACE project aimed at enhancing the conservation and management of European plant genetic resources. She was also the National Gene Bank in the Philippines and stewarded the beginning of World Work Package 4 at that time. Thank you for joining us. So we have very diverse people from very diverse regions, and this is an opportunity to listen to you on your aspects, your position of what needs to be done in the next 10 years, urgently, what needs to be done, how do we need to act, what is your proposal for what needs to be done now? And so I'll start with Karen first. Yes.
That works. Thank you very much, and we greatly appreciate this opportunity. So I'm from the Pacific Islands. We're a blue continent, not a green continent. 98% water and 2% land. So what happens on that land is critical. We are either farmers who fish or fishers who farm in the Pacific, and that still really applies to a large portion of the population. It's a cliche, I'm afraid, but we are also on the front lines of the climate crisis. It's daily and it's existential for the Pacific. And of course, biodiversity is entirely interlinked in that discussion. So on the vulnerability to climate change piece, and I think that's really critical, obviously sea level rise, extreme weather events are a risk not only to the biodiversity in the field, but also to the infrastructure of our gene banks. We need to think about that going forward and how we prepare for the inevitable. Literally right now, Fiji is being hit by a Category 3 cyclone. It's very early in the season. It's going to get worse, and it's going to get worse every year. So I'm sitting here just, hoping that the roof stays on the gene bank, literally. I'm confident it will. It's only a Category 3. If it was a Category 5, I might not be so confident. So for us, ensuring safety duplication is really critical and urgent going forward, and it's a high priority for us in the region. We've had people talk about data collection and management systems, and that's really critical as well. Excuse me. But we need to go beyond what we're currently collecting in our gene bank on data and information. And this doesn't have to be something that the gene bank does, but it is about how we network with the other information systems that are out there that add value to the data that we hold on our collection and our accessions. And an example, I have to say, is the Periodic Table of Foods initiative, which is a really interesting initiative that's going way beyond collecting the nutritional data of the important crops of the world. And that will be available open source. And for us to be able to find ways to link that sort of information into what we standardly hold on our collection would be really valuable for us to make decisions. Another critical concern of the Pacific is the NCD, not the non-communicable disease crisis. We have the triple burden of malnutrition, undernutrition, and overnutrition. And so our food resources, our genetic resources will be critical in combating that going forward. So the more we know about the nutrition of the varieties we hold, combined with how they will grow in our changing climate, whether they're salt tolerant or not, drought tolerant or not, and combined with the traditional knowledge that is not yet well documented for a lot of our varieties, is also really critical. Traditional production and agronomy practices, but also traditional uses for medicine and for food. That sort of information also needs to be linked in urgently because it's being lost to the data that we hold in a standard GeneBank collection. I think one of the other important things is around orphan crops and wild relatives, and I will totally adopt the name opportunity crops. I think that is much more appropriate. We need to look at the positive side of this. For us in the Pacific and for many countries in the less economically developed world, these crops are really critical for subsistence agriculture, but we need more research on them. Some of our staples, things like taro, yam, cassava, are not the most researched crops in the world, so there's also just basic big gaps there that we need to focus on. We need funding and we need commitment to do that. We need, to be frank, in the Pacific, plant breeders. We don't have any. We have no one, literally no one at the moment working full-time on plant breeding. So this is a huge gap in capability and capacity. So our region is small in land, it's also small in the number of people that we have. And that means a limited number of people working in this very specialized space. So again, it's the networks that we can work with globally to help build our capability and to help sharing and learning, which are really critical going forward. And to develop those and formalize those more will be very important for us moving forward. So in summary, to move us from the museum to the farm to the plate, it's strengthening those information ecosystems, strengthening the resourcing and the capability that will help us move from what is still really seen as a museum in lots of cases, I hate to say, but it is. Yeah. But also, as someone mentioned it a couple of times, the linking of the formal seed system to the informal seed system. The question I get asked most is from farmers is, how can I access what's in that gene bank? And then I have to say, well, you need to go through your government and it takes time, and usually they throw up their hands in dismay, and that's enough. So we need to find ways to make those links. I'd like to just call on one other thing, the reference to response to disasters. Also really critical for biodiversity and for our gene banks and for us to be positioned to provide support. We get asked every time there is a cyclone to provide planting materials. Our gene bank is tissue culture, that takes time. We need to be marrying what we have in our gene bank with field gene banks in regions, in the countries across the region, so that when there is a cyclone, they're in a position to quickly get planting materials from one island to another. I'm not sure how many of you are familiar with tropical cyclones, but if it hits a country, usually one part of the country will be hit and another part will be okay. So it's possible to do that at a national level. It can also help us support food relief in those situations as well, rather than the response being rice, which is not part of our traditional diet. And I'll leave it there. Thank you.
Thank you very much. Thank you very much indeed. So, because we're getting close to coffee, and I think people want to have that, 2-3 minutes, is that okay? 3 to 4 minutes maybe? Your priorities, where do you see us going? And then we'll give that into the session.
Thank you,
Thank
Janny. Good afternoon. I find myself quite in a unique position here, having recently transitioned to a much more sophisticated environment of IPK here in Germany. My eyes have been opened to the remarkable possibilities when strategic resources, cutting-edge technologies, and an extensive collaborative network come into play in the conservation of plant genetic resources. Conversely, where I came from, my initial work in a resource-constrained gene bank in the Philippines acquainted me intimately with the struggles, the severe challenges of conservation in a setting where government and institutional support is limited. In such countries, the hurdles of gene banking, be they financial, infrastructural, or technological, are not just more magnified. They define the daily struggle to safeguard these valuable resources. I believe it is understood among gene bank managers and curators that meeting the standards to keep their collections alive is crucial. They recognize the need to professionalize gene banking, to develop and adopt quality management systems, and to leverage technologies to valorize and rationalize their collections. However, the reality is one can only do so much. Many face the grim reality of not having enough, sometimes lacking even the most basic equipment. And so often they are confined by the limited resources at their disposal and compelled to operate on a crisis management basis. And yet, conserving the material, keeping their seeds viable is only half the battle. The challenge for gene banks is not just to exist and persist, but to evolve into entities of functional and actionable value. And so, for me, gene banks need to transition from isolated units to integrated players in broader agricultural and scientific landscapes. Too often, for some reasons, many gene banks function in silos, isolated from the very communities we aim to serve. And so gene banks need to be more accessible. The materials, the data, the information must be readily accessible. And we must actively engage with all the stakeholders, researchers, breeders, farmers, the academe, NGO, and the private sector. By doing so, gene banks can align their conservation strategies with the tangible needs of the sector. And such broadened collaboration can, in fact, help us, gene banks, to carry out our activities. This will not only foster and enhance utilization, it can also help us in addressing the bottlenecks. Academe, for example, it has been discussed earlier that academe can help us in doing the characterization. They can work alongside gene banks in decoding the vast genetic information stored within them. And also the private sector, the seed companies. We know that there are bottlenecks in regeneration in seed banks, especially in national seed banks. And so the private sector, the seed companies, with their financial and technical capacities, can actually help us in this more resource-intensive gene bank operations. Of course, it's worth acknowledging that this must be built on mutual trust and respect and adherence to all the policies and laws. But yeah, NGOs, given their community-centered operations, can help gene banks, establish the gaps, foster the relationship between gene banks and the grassroots stakeholders, ensuring that conservation is informed by on-the-ground realities. And another collaborative approach would be citizen science, by involving local communities. In genetic resource documentation, for example, we do not only enhance our data sets, but also foster a sense of collective ownership. And finally, what I would like to address as well is through this active engagement with all the stakeholders, gene banks can hopefully effectively influence policy. Actually, one of the reasons why we lack funding is that because we lack the policies relevant to PGR. And so with joint efforts and collaboration, I believe that gene banks can nudge policymakers to craft national strategic action plans. Actually, it's my dream, it's my hope for my home country to come up with a strong national strategic plan, because The National Plant Genetic Resources Laboratory in the Philippines was established 47 years ago, but the development of a policy and a strong strategic action plan is still pending. And so it is my belief that with consensus effort and strong collaboration, purposeful collaboration, stakeholder engagement, and policy advocacy, we can unlock the transformative potential of gene banks, especially in resource-limited settings. Thank you.
Very eloquent. Lise?
Thank you. There have been said so many wise words today already, and I think I'd take another path, not to echo everyone. The only thing I would say, which is an echo, is that we really need gene banks to be able to have resilient agri-food systems. And we are talking about critical biological infrastructure. I think this is sometimes forgotten. But what is a well-functioning gene bank? First, we have to think about what is a gene bank, because we think we all have a picture of what is a gene bank. But if you look around, you visit fellows, you see that some places a gene bank is a seed storage, or it's a seed distribution center. Other places, gene banks are well-functioning research centers. They do genomics, phenomics. They do a lot of projects. They combine the knowledge of the seeds. They have regenerated 100% of their seeds, and they have 99% in the Suva Global Seed Vault. So they come in different functions, and they're locked up in different ways. Some have a very clear line of command, others are hidden in universities, and if you walk down the second building in the third corridor, you find it. So there are people taking decisions on gene banks, and gene banks, we have talked a lot about why do we need gene banks and what can they contribute to. But what kind of gene bank does a certain country or organization want? I think this is a really, really critical issue. It demands leadership. I was asked in an interview half a year ago, if you have all the funding that you could ever dream of, what would you look like in 5 years? That was mind-blowing, because just thinking of that. So I just think maybe we should start by having a dream, a clear purpose, and a direction for where are we going with my particular gene bank. There are thousands of experts telling what the gene bank should do, but if it's not what the gene bank wants to do, or the decision makers, you won't get there. So I made a dream, okay? A long-term, well-funded gene bank, not just funding for 3 years, funding for 20 years, because we work for decades, in 100 years perspective. Yes. We don't work in a 2-year term. Come on, perennials, we won't even have the seeds, right? We want great leadership. We want somebody who really takes responsibility. Very often, I have colleagues where they are 2 persons and they are biologists and they don't even have 1 drop of leadership within them, but they're the best curators. How should that gene bank develop then? So we need leadership. We need efficient work processes. We need equipment. We need technologies. We need motivated staff. And that's what we all have, actually. Motivated, passionate staff, because we have a true purpose, a true star where we're guiding us. This dream would also, we can deliver all orders at the same time. We don't have to wait for multiplication. We don't have to dig it out somewhere or we can't find it. So we want to participate in research, and we want to take part of those research projects, not just deliver seeds. And we are in AI projects. We are doing phenomics, genomics, and we are not in a silo. We're working across. Isn't that just a wonderful dream, right? Okay. But there are no quick fixes here. And there are not one gene bank who looks like the other. They might be similar, but there are some trends. For the first time, Europe has a European Gene Bank Manager Network. And I have the pleasure of chairing the first meeting ever for the European Gene Bank Managers on Thursday. And beforehand that meeting, I made a small survey and I asked the gene banks, and there are 60 people in this network, which is much too big, but that's what it is. I asked them, what do you need? And based on that, and based off my travels around the world, I have some trends here. And I think that goes for, most, that whenever you're a seed storage or you're a full-fledged research center. We need funding, long-term funding. And we need relations. We need to have close relations to our stakeholders, and we also need to have something to come with. If we cannot bring knowledge, if we cannot bring more than the seeds, we will have difficulties in getting the funding, because there are no funding for operations. Funding has to be to something much more that you can relate to. So in that triangle, the better relations, the better knowledge you have, the more you can put it into play, the better your options are for enter into a consortia and get some money. And I'm talking on experiences here. We need databases. We need data management. The information on the seeds is as valuable as the seeds. And it's not enough to have it in an Excel sheet and the computer break down and you lost all your information. We need to transfer that, and we need to, those who are our users, they need to be able to find the information. They can't wait to someone digging it out. They are busy people. We need capacity building, and we need to share that. Everything from, we're using this seed counter in the seed lab, to looking into seed technologies, how we store genomic data, anything, or how we regenerate, or our quality management system. How have we built it up, or do we have one at all? What information have we put in there? Collaboration is key. Everyone has said that, and it's truly key. But if you don't have the resources, you have really small collaboration because it takes time. You rather want to make sure that cauliflower produce some seeds than going to a meeting with someone else. Awareness raising. I don't know how many parties you have been to, but every time I have someone sitting beside me and they said, "What are you doing?" and I tell them, I said, "Why have nobody told me about that?" I said, "Well, I don't know." So, but if you don't know the value and importance, like this event is really making some buzz. All of you here know what gene banks are, but if everybody goes from here and tell more about it, and we go on social media, we will put it higher on the agenda, right? I would say that action calls on all levels, but listen to the gene banks and see what are their capabilities and what are their wishes, what are their dreams. Is it the seed storage, is it a research center, is it to being able to do breeding or whatever it is? That must be the center of it. And then they can learn from colleagues and experts and all this. But if they're not really interested in learning that, we are using our time and money and efforts on the wrong things. Thank you.
Thank you very much, Lise. Two minutes. Sorry, but 2 minutes and you have to go. Yes. Thank you. Yes.
Distinguished dignitaries and ladies and gentlemen, good afternoon. I salute everybody, and I want to tell you that I'm from Nigeria, and Nigeria is a new entrant into the treaty. So, yes, we signed the treaty in 2002. By the grace of God, were able to ratify in 2022, 20 years after. So you can imagine what that does for us. And as a Genebank, of course, aside the great awareness, which my colleagues has emphasized on, truly in the house here, mixtures of politicians, policymakers, and so many others, many of us don't even know the functions and the management of Genebank. And of course, it is one of the key things for us in Nigeria, after being a member and being fortunate to be here, and as well as the next week meeting in Rome, we would love to tell the House that awareness is one of the key. To letting people to understand the functions and management of Genebank. My colleague has said here, it's not a museum. It's not a place where we just store things. They are living things, just like us. And we need them to drive the agri-system. And of course, breeders, what they need is the raw materials. And where is the raw materials? In the Genebank. And of course, the Genebank does not just go there and make collections. We collect, and preliminary assessment by evaluations are done, so that when breeders now need materials, they don't need to reinventing the wheel. You may just come, I need some accessions of materials that are dry, for drought, or for this, or for that. At least from your kitty, you can just, hand down, and of course, they cannot take it from there. So aside all this, of course, infrastructure, highly needed, capacity building, highly needed. But presently, for Nigeria, because of some of the challenges we have by way of some insurgents and thereabout, there is need for aggressive exploration and collection for so many of the places that the Genebank has not been able to go to make valuable collections of our materials. And of course, Nigeria, as many of you may know, diverse with several crop species. For us, we've been able to manage to have about 11,000 accessions of about 38 crop species. And this, with the population of Nigeria, which by World Bank rating is over 200 million now. So how do we feed ourselves? And of course, the global vote that is also helping to making sure that this material globally is being kept. How do we contribute into that? So for us, we believe that with all these, I've been able to, by way of Janny telling me 2 minutes, I think I have been able to critically tell what I believe for the future should be done in order to consolidate some of the things we're doing. Thank you very much.
Thank you. Thank you, Tony. Apologies for cutting into your coffee, but I think you will agree that we had to listen to everybody, and we had to give each person the time to express their thoughts, their dreams. Excellent. I thank you from the bottom of my heart for bringing such a strong presentation, all of you. Thank you so much for giving us these thoughts, and thank you to all of you also for hanging in there. We'll see you soon, and hopefully you'll enjoy your coffee now and have a time to discuss what we presented. Thank you so much.